As we discussed in previous module that there are three dilemmas when we replicate DNA. There are three issues that we need to sort out. So how does cell sort out these three issues? Let's look at the first dilemma. We talked about that, the lagging strand. We said that DNA molecule is separating in smaller, is gradually separating. The two DNA strands are gradually separating. The leading strand can be synthesized continuously because it is being synthesized in the direction in which the two DNA molecules are separating. So no problem there. But the other strand, the lagging strand, can only be synthesized in the area where DNA has separated. And this strand, as we said, is sort of being made in the wrong direction. Because as you can see here, there is the 3' end of the template DNA. Complementary strand would have a 5' end because the DNA is anti-parallel. So it's pretty straightforward to make the leading strand. This is the leading strand. However, when it comes to lagging strand, when the primer is made, the orientation of primer would be that it would be 5' end would be here and 3' end would be here. Because if we extend this 3' end, it will, when it becomes complementary to this base over here, it will be also 3' and it will still maintain the anti-parallel feature of the DNA. So the lagging strand is made in smaller units. As the DNA space becomes available, primase will lay down the RNA primer. That primer will be extended by DNA polymerase DNA polymerase 3. And after it has done its job, it will stop. Again, when the helicase opens up more region of DNA, a new primer will be made and DNA polymerase will again extend this primer. So this is how the lagging strand is synthesized. However, there are other issues regarding making the lagging strand. Primarily, it is the RNA primer. We need to get rid of RNA in the DNA molecule. So the lagging strand is sort of hybrid between DNA smaller pieces and RNA smaller pieces, which are the primers. So how do we do that? So there's another enzyme called DNA polymerase 3. By the way, there are 3 DNA polymerases in E. coli and 14 in eukaryotes. We are looking at the eukaryotic system. So we have 3 DNA polymerases, 1, 2, and 3. 3 is the one which just synthesized our leading strand, and it has also synthesized the lagging strand. The DNA polymerase 1 comes in and removes, it has 2 activities. It will remove the RNA primer, which was made initially by primase. It will remove that primer and also extend the DNA. So the now 2 DNA, as you can see on the screen, they are adjacent to each other. However, there is a gap between these 2 DNA fragments. We need to seal that gap. That is done by another enzyme called ligase. Ligase will come in and it will establish the phosphodiester bond here. So now our DNA is a single molecule and it is stretching. So we have removed the RNA primer and we have made a continuous DNA strand, which was initially chopped up, which was partly RNA, partly DNA. We will continue our efforts to explore the DNA replication in the next module, and we will look at the dilemma number 2 and 3.